So, what if we could find a unique fingerprint for a disease as complex as multiple sclerosis? Today, we're diving into some incredible new research that might just make that possible, and it could totally change the game for diagnosing MS.
Welcome to Brain and Data. This is where we break down the amazing science that's pushing the boundaries of what we know about the brain. So, where does this all come from? We're looking at a huge study from researchers at the Max Planck Institute and the Technical University of Munich, which just came out in the journal Cell. And trust me, this isn't just another scientific paper, it's a potential revolution in the making. Let's cut right to the chase. Here's the headline from the study itself. They've developed a 22-protein test that nails the diagnosis of MS, especially for those really tough, confusing cases. For so many patients, this could be an absolute game changer. Okay, so here's how we're going to break this all down. First, we'll look at why we so desperately need a new test in the first place. Then, we'll get into the incredible tech that made this research possible. After that, we'll unpack the 22-protein fingerprint itself. And finally, we'll talk about what this all means for the future. To really get why this new test is such a big deal, you first have to understand just how difficult and honestly frustrating it is to diagnose MS right now. You know, for a lot of people, getting a clear MS diagnosis is this long, agonizing journey. Get this, about 1 in 7 people are actually misdiagnosed. This can mean years of delays in getting the right treatment, or even worse, it means people are getting powerful drugs with serious side effects for a disease they don't even have. It's a huge problem. For decades now, the go-to lab test has been to look for something called oligoclonal bands, or OCBs, in a person's spinal fluid. It's basically a sign that the immune system is fired up inside the brain and spinal cord, which is a classic hallmark of MS. But here's the kicker. For about 1 in 10 people who definitely have MS, this critical test comes back negative. They are OCB negative. Their immune system is still causing damage, but the test, it just can't see it. And for this OCB negative group, things get even harder. Their chance of being misdiagnosed actually doubles. They're left in this awful diagnostic limbo, all while the clock is ticking. This is the exact gap that this new research wanted to fill. So how in the world did these researchers even begin to tackle this? Well, they didn't just look for one new clue. They took a completely different approach, one that was only possible because of a massive leap in technology. Think about it this way. Instead of looking for a single clue at a crime scene, they decided to analyze the entire crime scene. They mapped out the whole universe of proteins floating around in the spinal fluid, that's called the proteome, to find a complex signature, not just a single footprint. Now, to find a signature that subtle, you need a mountain of data. And they got it. They analyzed samples from over 5,000 different people. I mean, that scale is just unheard of for this type of work. It's way, way bigger than anything that's come before. And this image here really lays out their game plan. You can see they took thousands of samples from people with all sorts of different neurological issues, mapped out all those proteins, and then used that massive database to find a specific pattern that screamed multiple sclerosis. So how do you even process that much information? Well, you build a kind of futuristic, high-tech assembly line. They had robots preparing the samples, then a super-fast machine called a mass spectrometer analyzed them at a crazy speed. They went from doing 12 samples a day to 100. And then they let powerful machine learning algorithms loose on the data to find the hidden patterns. Okay, so after all of that incredible work, all that data, all that tech, what did they actually find at the end of the rainbow? Well, they found the fingerprint. The AI crunched through literally millions of data points, and out of all that noise, it pinpointed a unique combination of 22 specific proteins. It's the levels of these proteins, when you look at them all together, that act as a highly specific signature for MS. I mean, this is a gigantic leap forward. We're going from looking for a single piece of evidence, which we know can be unreliable, to doing a full forensic analysis using 22 different data points. It is just so much more robust. And hey, the results really do speak for themselves. This graph shows you exactly how well the new test works. That blue line soaring upwards, that's the new 22 protein model. The green one down below is the old method. You can see that huge jump in accuracy. And look, it performs brilliantly for those tough OCB negative patients. It shines exactly where the old test fails. Okay, a discovery like this in a lab is amazing, but it doesn't mean anything until it can actually help patients, right? So what's the path from this scientific paper to your doctor's office? This is the really smart part. The researchers didn't just stop at the discovery. They took this complex finding and they turned it into a practical, targeted test that just measures those 22 key proteins. It's a format that's actually designed to be used in a normal hospital lab. So let's just recap what they've managed to do here. They discovered the panel in a massive group of people, they proved it worked in a second independent group, and they built a test that's ready for clinical use. They've checked all the boxes. The next step is to test it out in multiple hospitals to get it ready for prime time. This whole thing leaves you with a really powerful question. We've just seen a blueprint for creating a super accurate test for MS, but the bigger picture here is massive. Could this exact same approach, this combination of huge data sets and smart AI, be the key to finally cracking the code for diagnosing other complex brain diseases and turning diagnosis from an art into a precise science?